So, after the sensitivity range in the performance factors, another very important aspect is the accuracy. The sensor accuracy has to be, in most of the cases, better than plus minus 5%. So, accuracy also reflects the repeatability, or the kind of parity between the measurements that the sensor makes with time if you have multiple readings. Okay. Another very important aspect of performance, which has to be kept in mind, it's a kind of intangible aspect, is the nature of solution or the variety of solution that the sensor can scan. So, when you design the sensor, a very important aspect to take care of is that whether the sensor can withstand various conditions of pH, temperature, ionic strength, etc. And so, essentially the sensor, when you talk about designing the sensor, it has to be designed for its operability, its ability to get operated in varied conditions of different pH, temperature, ionic strength, etc. Okay. So, that is another aspect, a performance factor aspect of a particular sensor. Another very important aspect which makes the sensor really give a commercial angle is its performance time, or response time. So, for biosensors, typically the response time has been found out to be a little longer. It takes about 30 seconds or more. In this context, I would also like to say that in electrochemical sensing operations, this sensor time becomes very high, because of the fact that you have to recondition the electrodes, the sensing electrodes, and that is a very time consuming process. So, that essentially, although it is not a part of the actual response time, comes in the overall time of sensing of such an electrode. So, if you are having a real time process where there is a continuous monitoring of the analyte of interest with time, it is a very critical aspect, that what is the response time of the sensor, how soon can it process a chemical information into an electrical or otherwise optically readable signal. Another very important aspect, it is also a part of the performance factor, is the recovery time of a particular sensor. So, I would like to define this as the time elapsed before the sensor is ready to analyze next sample. Okay. So, essentially it is the time between the readouts. So, if you have two or more certain readings that you want to take with the same sensor, how much amount of reconditioning time is needed, so that you can get the sensor ab initio, so that the sensing can start on a very accurate basis. So, that is an important aspect of the performance factor of the sensor. So, if you have a recovery time of more than a minute, more than a few minutes, then really it does not make sense in real time operations anymore. And very important aspect, another very important aspect, is the working lifetime of a sensor, like things like stability of the selective material. It defines these kind of issues. In case of glucose sensors, as I was illustrating in my last lecture, this enzyme glucose oxidase is a very finicky enzyme. It can only have a defined shelf life. So, if these kind of moieties, which are essentially recognition elements, they change their behavior with time, what good it is to the sensing aspect. So, the working lifetime is a very important factor that needs to be considered if you want to design good sensors. Let us actually now go to the next level and try to find out that this whole kind of race to miniaturize, starting from let us say the microchips. Okay. Why really it is important? What are some of the reasons for miniaturizing let us say sensors? Okay. So, the justification can be provided by looking at several aspects. One aspect could be that as we had talked in the scales and sizes, the reduction of sensor element to the scale of the target species would actually give a higher sensitivity for detecting a single entity or a molecule. So, it is a very important aspect. So, you are talking about the particular detecting element to be of the size of a single molecule or a single moiety. of interest. So, when the size comparison is there is automatically a higher sensitivity of detection of such a process. Another very interesting aspect is reduced reagent volumes and costs of operations. Some time back I was mentioning about this process of polymerase chain reaction, where you are artificially trying to amplify a small segment of DNA, and it's a fascinating process, but unfortunately it is a very expensive process too, because it uses some of these chemically isolated base pairs, some enzymes, some ionic buffers, etc., which gives the whole reaction cost a different dimension altogether. So, therefore, more number of PCR runs essentially is at a cost, is at a more money spending issue. Okay. So, therefore, it is important that if we can design a sensor in a manner which can do a micro PCR, that means you take a small volume where, let us say, the overall volume is about 1 microliters in nature, and then you have these different components which go into the reaction, probably of the order of picoliters. So, in that case, the cost, the overall associated cost reduces. So, one of the reasons why miniaturization is important is that the reagent volumes used for chemical analysis automatically reduce because of the size factor. Reduced time to results due to small volumes is a very important aspect. And as we know that when we talk about concentrations of different moieties within a solution, there is an aspect of diffusion which comes into picture automatically. That if you have a gradient of concentration, there is always a tendency of molecules to diffuse and homogenize the particular medium. So, if this overall volume over which we are talking is very small, then the diffusion mechanics is much more rapidly taking place, and as a result of which the homogenization from which the sensing activity would start really is much quicker. Okay. So, this has its effects in terms of response time of the sensor also. That miniaturizing a sensor or miniaturizing the volume of fluid which is to be sensed essentially reduces the time to homogenize, so that you can have an accurate readout much faster. Of course, portability is a major issue. These days there is a concept of bedside monitoring of a patient, wherein we also know this whole area as point of care diagnostics, where we take a small sensor to where a patient is, and really the sensor does a job, its job on a real time basis, just by just continuously monitoring the patient's health, and such sensors have to be miniaturized, because otherwise if you move the whole laboratory, how is it possible really. So, the concept of lab on chip really emerged because of the need to make things more portable, so that they can be more delivered at the point where it is needed essentially. So, amenability to portability is a very important aspect why the sensor should be miniaturized. So, point of care diagnostics essentially, which we talked about. I would just like to share with you that nowadays in advanced healthcare schemes there are small ring like sensors which they would just put around your finger, and they can monitor your things like blood glucose level very easily on an optical basis. So, any patient who goes into an emergency care essentially is given such a ring to wear, and the ring has a perfectly wirelessly communicating system which would give information about the content or the hemoglobin, the glucose content of the patient from time to time. Okay. So, this is point of care. This comes very close to where the patient is placed actually, and does its job on a continuous basis. Miniaturization is also helpful for multi-agent detection capabilities. What essentially it means is that if you have more than one agents that you want to detect, and if you really want to detect each agent on a very small basis, you could densely integrate several such detection protocols on a small particular chip. And that is the essence of microchip technology essentially, that you can on a very dense networked basis do multi-agent detection on a very small area. And the very important, last but not the least, why miniaturization is needed, at least in some of the reasons which appear to be the most prominent ones, is the potential for use in vitro as well as in vivo. So for those of you who are new in this area, in vitro essentially means outside the human body. Okay, so the sensor can do its job outside the human body. In vivo, on the other hand, is inside the human body. So all these real-time sensors that we have been illustrating from time to time are essentially in vivo sensors. You implant a sensor onto the human body and it keeps on monitoring the health rapidly, and it keeps on giving data rapidly. So that is an in vivo sensor. So miniaturization is very helpful for in vitro as well as in vivo sensing. Okay, so one of the reasons is that if you are talking about an implantable sensor and the sensor is of the size of a pen, can you imagine the amount of pain that the patient is inflicted upon if you want to locate this sensor somewhere within his body? Okay, so the smaller the sensor is, the better in that case, because it can cause a painless delivery of the sensor to the target where it can detect, or it can start detecting. So again, we have been talking about lab on chip devices. I guess you have a fairly good idea about what biochips are. So let me just definitionally kind of outlay what this technology is all about. So lab on chip is a term for devices that integrate multiple laboratory functions on a single chip. So very obvious, the definition that you have several functions in a laboratory which are kind of integrated together to do some detection. So lab on chip is something that brings all these functions to a very small single chip. There are enormous applications of lab on chip as on date. There are several commercially available protocols too. There are applications in medicines, pharmacology, pharmaceuticals, for food safety, so on and so forth. And some of the advantages that such devices offer on a commercial basis is that they are fully integrated and essentially without, or maybe with very less, I should say, human intervention. Okay, so it is a very clean process. You don't have chances of contamination just because of multiple handling, as happens very often in the laboratory. They are highly sensitive in nature because of the miniaturized form of these chips, as we have been often on talking about before. They are extremely rapid, so the time of response is very less because of all those homogenization aspects of the analyte, and being a small volume, the diffusion restrictions are automatically minimized, so on and so forth. And then this other aspect of cost performance product. So essentially these are low cost, high performance devices. So that's why they are really high utility. The lab on chips are really a high utility kind of devices. Okay, so some of the companies selling these technologies across the globe, to name a few: Nanogen, Caliper Technologies, Affymetrix, Clara Technologies, so on and so forth. And essentially these two companies, Nanogen and Affymetrix, they are selling or catering to the whole diagnostics industry by selling gene identification platforms. Okay, and we'll be discussing in details about these as we go along our lectures, as to some of the strategies and mechanisms that these manufacturers use to identify this sequence of base pairs on a single DNA molecule. So in general, there's a known fact that if you look at all the commercial applications of all these lab on chip systems, more or less a majority, I would say almost 80-90% of the products which are currently available are in this integrated gene analysis area. Okay, and there are some products which have been developed in the immunology, like Aclara has a product, or this Caliper Technologies has a product wherein immunochemistry is used for identification of cells or biological moieties. So that's in a nutshell what the status of lab on chip techniques are. I would now like to illustrate a very important area which has come off late because of the development of these tools, and this is also known as nanobiology. Okay, So the MEMS technology, in fact the miniaturization technology, is very useful for studying certain aspects of single cells. Okay. So here in this particular case, as you are seeing, this is a controlled microenvironment in a biochip where you have a kind of it's like something like a, let's say, a nanowire, okay, between two posts, and you have a silicon platform here. It is like a MOSFET device, okay. So essentially whatever is going on in terms of chemical changes in top of this region gets converted in terms of a surface potential, and if there is a charge transfer process which is happening in this particular region, the surface potential change would result in a variation in the drain source current in the MOSFET device. And so therefore it can be a very good identification basis for what is going on in a single cell which is immobilized into this area. So you give a stimulus to the cell and see how the cell performs. Now how is it important? This is very, very interesting that actually there is an increased requirement of understanding of the physiology of a single cell in the presence or absence of its identical other cells, and so there is a lot of signaling mechanism between let's say more than one cells growing on a certain area, which results in a totally different behavioral change of a cell of interest. So cell physiology essentially nowadays focuses on to these molecular events which take place in the presence or absence of a group of other cells. One more interesting factor is that the way that proteins are produced, and I'm going to actually introduce this topic a little bit doing a basic biology, that what happens in a cell. Okay, so there is a certain programming aspect of a particular cell which we have to really read if we come to such a mechanism. So as we know that there is this molecular information stored within the DNA of a particular cell. Okay, so this information is a basis of giving what kind of proteins the cell produces. Okay, this area nowadays is increasingly going because proteins essentially are a very important basis of the state of health of a certain cell. Okay, so there is a process called transcription and translation within single cells, and it's a huge nano machinery which is at work. There is a tendency of this messenger RNA which is a compressed code essentially encoding a certain area or region of the gene that we are talking about, and this RNA actually comes all the way from the nucleus into the cytoplasm, and through the cytoplasm it goes to something called an endoplasmic reticulum, which is also the protein warehouse of a cell, where there is a coding which happens on this mRNA, and the base pairs of mRNA are converged rapidly into a sequence of amino acids, which is essentially the protein which is being produced. So this is a fantastic process which is going on. So nanobiology again studies very closely these signaling effects within the cells, which asks the cell to code a certain region of the gene based on the impulse or the response that it has from an environment. And such studies can only be possible when you make the sensing system doing these studies on a very, very highly miniaturized basis. Okay, so in a nutshell, what I would like to say is, the increase in these concepts of miniaturization has resulted into this fantastic field of nanobiology to get developed, where analysis of single cells and study of their function in real time can be accomplished. There can be an increased understanding of the signaling pathways inside the cell. Some of them we have been mentioning off and on. Basic cell functions such as how the cells differentiate in the presence or absence of its own types around it, the way they reproduce, okay, the way they apoptose or die after a certain time, and their implications on various disease states, they can be understood on a very molecular level. And essentially the focus of the post genomic area and systems biology, some of these newly emerging fields are in this area more often. Cut. So nanobiology definitely is probably one of the areas of interest which automatically gets cropped up because of this micro systems technology. So this pretty much brings us to the end of this lecture, and next time we will be seeing how microfabrication tools can be used to realize things which are small and miniaturized, which could do some of these jobs that I have been mentioning over the last 2 lectures. So probably we will discuss a little bit of microfabrication related processes in our next lecture. Thank you.